Hello and welcome to lesson 4 in the iCow soil biology course. Today we're going to be learning about fungi. We'll start by reminding ourselves about the soil food web, which defines for us who eats who in our soils, who's dependent on who, and how collectively all the creatures in the soil stabilize a healthy soil so that we can grow healthy crops, have clean water, and have a healthy overall planet, environment, and healthy livestock. The soil food web is divided into trophic levels, with the first being the photosynthesizers. And the second trophic level are the decomposing mutualists, pathogens, parasites, and root feeders. Our last lesson we learned about the bacteria, which are in this second group, and today we're learning about fungi, which are also part of this group. So, in her book, For the Love of Soils, Nicole Masters describes fungi as being the organisms holding the fabric of soil life together. Michelle Masters is an independent agroecologist, a systems thinker, and an educator. She says that fungi are our underground livestock. Actually, all our microorganisms under the ground are our underground livestock. And that what sets fungi apart from other kingdoms of life is chitin, which is a long-chain carbohydrate found in their cell walls. As individual hyphae, that is, individual strands, they're invisible to the naked eye. They're 2 to 10 microns wide, but as they adhere and bundle together, they become the visible mycelia, the structures that you can see on decaying wood. So those are the mycelia. The actual independent strands, the hyphae, these are the ones that we see under the microscope. And these are 2 to 10 microns wide. Our bacteria, in comparison, are only 1 micron wide. So they're significantly bigger than bacteria in terms of width. But of course, very, very different shapes. Nicole gives us a good visual reference by further describing the soil fungi as being like the cement and stones that build our homes. They're the vital part of the foundation in the soil structure. They're also, though, the neural network of the soil, sending information across thousands of acres, communicating insect attack, instigating plant defenses, and also sharing resources. Estimates point to at least 70,000 species of soil fungi across the world. These include yeasts, molds, and mushrooms. However, this figure is just an estimate. Many more are unknown. You can see that they're almost like a drip irrigation system under the ground, connecting different plants, bringing different benefits in terms of different resources, connecting plants in a neural way. If you imagine our own nerve system, if somebody bangs your toe, you feel it and pull your foot away. You don't just feel it in your toe, you almost feel it across your body. And this is almost how the underground are connected in terms of their nerves. So you can imagine what happens when we go in there with a djembe or a plow and we dig all of this up. We break the communications, we break the spread of resources, and we actually do more damage to the plants growing around and above the ground than we realize, because we're cutting them off from these fungal networks. Soil fungi groups can be categorized as having 3 major functions in the soil: mutualists, saprophytes, and pathogens. The first of this group, the mutualists, offer an array of benefits to plant and animals. There are examples of animals that choose to partner with and even farm fungi. Can you imagine? There are animals that farm. These include leafcutter ants, stingless bees, beetles, and termites. So next time you look at a termite mound, think about this. They cultivate the fungi in a fungus garden, comprising a few hundred fungus combs, structures built from chewed up grass and wood, and inoculated with fungal spores. Much the same way when we try to grow mushrooms, we do the same thing. Each year, these fungi in the termite mounds produce a crop of large mushrooms. In some areas, these mushrooms are known locally as omojoa, which are highly prized as a delicacy. So next time you're looking at these mushrooms around a termite mound, just imagine, like us, There are insects that are farmers. That's just so cool. Lichen and algae require this mutual relationship with many species of fungi for their survival too. If you look on the trees around you, you'll see these little things called lichen. Next time you see them in your environment, take a very close look to see if you can see their fungal support system. Lichen are a complex life form that is a symbiotic partnership of two separate organisms, a fungus and an algae. The dominant partner is a fungus, which gives a lichen the majority of its characteristics from its shape to its fruiting bodies. We have them all around us. Mycorrhizal fungi, micro meaning fungal and rhizal meaning roots, are mutualists which live on and in plant roots, creating a symbiotic relationship between the plant, fungus, and their bacterial partners. Remember, down there in the root zone, in the rhizosphere, are the bacteria we've learned about. And now we're seeing that actually there is a relationship between the bacteria, the fungi, and the plant roots. The mycorrhizae reduce plant stress. They offer root protection and water nutrient uptake in exchange for carbohydrates, carbon, energy, and fatty acids. Remember, we said earlier that what sets fungi apart from other kingdoms of life is chitin, which is long-chain carbohydrate found in their walls. Well, they need to get this somewhere, and they get it from the plants. So they give the plants what the plant needs, and the plant, through photosynthesis, brings down those carbohydrates, carbon, energy, and fatty acids, and allows the fungi to access these so that the fungi can grow. Their hyphae are much finer and can travel much further than plant roots. In fact, plants that have fungi down there in their roots increase the surface area of access to water and nutrient uptake by up to 40 times. So who wouldn't want some in their farming system? Mycorrhizal fungi are a critical and significant component of healthy soil ecosystem, making up as much as 1/3 of the microbial biomass in healthy soils. Over 90% of plant families of 250,000 plant species have this mycorrhizal relationship, supporting healthy, resilient plant growth. So we really, really depend and need these mycorrhizal fungi, this special group of fungi connecting underground. Saprophytic fungi are the largest group of macrofungi responsible for breaking down and recycling dead plant and animal material. These are the fruit bodies you see on dead trees, litter, animal bones, even on feces. Saprophytic fungi release enzymes to break down and digest the lignin, the cellulose, or the chitin in this material into simple soluble compounds that can be absorbed by them and by plants as nutrients. In so doing, they play a vital role in reducing the accumulation of dead organic material and in recycling essential nutrients, particularly carbon and nitrogen. Without these digestive activities, forests would disappear under a mountain of logs and leaves. And then we come to pathogenic fungi. Pathogenic fungi are fungi that cause disease in humans or other organisms. You hear me talking about fungi or fungi, they're the same thing. Approximately 300 fungi are known to be pathogenic to humans. Many more fungi are known to be pathogenic to plant life than those of the animal kingdom. The study of fungi pathogenic to humans is called medical mycology. We've all heard of athlete's foot, candidas, and ringworm. And in plants, we're all familiar with fusarium, mildews, rusts, botrytis. Many of the fungi that we learn about are the pathogenic ones because we try to control these, but we very rarely learn about those that we depend on for symbiotic relationships. When we go and address some of the pathogenic fungi in our agricultural systems, we kill the good fungi as well. The same way when we use pesticides to kill some insects, we kill the beneficial insects too. Mushrooms are the fruiting bodies of some fungi. So next time you're looking at mushrooms in the shops or in your garden or anywhere, just know that they're the fruiting bodies of some fungi. So let's look at how they reproduce. Most fungi reproduce through asexual reproduction. That means that they don't need a female and a male connection, but rather the young get all of their genes from a single parent. Although fragmentation, that's pieces, fission, that's joining, and budding are methods of asexual reproduction in a number of fungi, the majority reproduce asexually by the formation of spores. Now, where this is very interesting is also looking under the microscope. We can see, when we see fungal spores, we can actually see the mycorrhizal fungal spores. We can actually even see the pathogenic fungal spores, which give us an indication of what is happening in our soils, or what is about to happen, or what can possibly happen in our soils under the conditions that these spores would actually come alive. And what we do know as well is that spores can stay in the soil for tens of thousands of years. Here we can see a variety of fungal spores as seen under the microscope. As you can see, they come in a variety of colours and shapes. Some of them there are 10 microns, some of them are 50 microns. So the spores are quite large quite wide compared to the actual fungi that they produce, or the hyphae that they produce. These pictures of spores were taken in our lab. The first is of wild mushrooms that sprouted after the rains, and the second two were in compost and a vermiculture sample. Their large size and dark colour indicates that they're from good soil fungi, and perhaps even mycorrhizal fungi. Fungi are everywhere. Here we can see fungi in a packet of tomato juice. We looked at the green mold on top of the fungi under the microscope and saw that in fact it was green spore. We need these fungi. This indicates to us that this tomato paste is not good enough for us to eat. So fungi are really great in indicating to us that some things might be dangerous for us to eat because they've reached their expiry date and perhaps are even producing toxics now with other organisms that may be in them. So when you see fungi growing on things, we know our automatic reaction is, oops, we need to chuck this out. So look at fungi as being your friends. As we've seen in some of the pictures above, pathogenic fungi are not desirable. When we look at these under the microscope, we can see some very obvious differences. Colour, diameter, and cross walls are quick indicators of good or pathogenic fungi. On the left there you can see what we consider to be good or beneficial fungi, and you can see that the colour is a brownish colour, the diameter is quite wide, we've said from 2 to 10 microns, and we can also see the cross walls. We call these cross walls septa or septae, and sometimes you can also even see oxalic acid crystals on the outsides of the fungal walls. This is where the fungi are changing their, we call it the cation exchange, where they're changing their ions or attracting negative or positive ions onto the fungi wall, and in order for the fungi then to absorb those nutrients using their enzymes and absorb those nutrients into the fungal, into their systems, and then use those to pass on, move on, grow with whatever it is that they want to do with them. On the left side there you can see the non-desirable soil fungi. So typically they are colourless under the microscope. Their diameter is quite narrow in comparison to good fungi. They are approximately 1.5 to 2.5 microns wide, or to 3.5 in some cases, but typically about 1.5 to 2.5. They may have cross walls or may not. Some of them have periodic or irrationally spaced septa or cross walls, whereas with the good fungi, they're normally if you look at them, they're the same distance, equidistance apart, but they're not like that most of the time in the bad fungi. When we look through a sample through our microscopes and we're looking through your soils or compost, we're looking to identify which fungi are present. If you look closely at this one, you can see a tiny fungal strand right in the middle of this picture. This is only at a 40 magnification, so 40 times larger than it is. But to absolutely identify it, we would need to increase the magnification all the way up to 400x. That's 400 times. And here we can see some good fungi again. The nice brown color, clear and evenly distanced septa, and the picture down at the bottom, you can actually see the little granules of the oxalates on the outside of the fungi. And then here are some pathogenic fungi. They're narrow, they're messy, they're transparent with irregular septa, and they can grow really, really quickly. We know that fungi are great decomposers. They therefore hold nutrients locked up in their structure, and they also predate or are predated on by other organisms in the soil food web. And these organisms cycle these nutrients back into the soil in plant available forms through the poop loop. So again, looking at this picture above the sun there, we've got living plants that break down into dead organic matter. That dead organic matter is then taken up by the second of the trophic level in the soils, the decomposers, the saprophytic fungi, the aerobic bacteria, and they in turn accumulate nitrogen. And then these are predated on by protozoa, amoeba, flagellates, and ciliates that poop, and others actually, and micro and macroarthropods. But when these excrete their poop, they put back those nutrients that they have got from the bacteria, from the fungi, they put it back into the soil as plant available nutrients. So that's how the poop loop works, and that's how the nutrient cycling works inside the soil. So you can see why it's absolutely beneficial. If you just look at this picture and imagine that you didn't have bacteria or fungi, there would be that gap. There wouldn't be proper nutrient cycling. Or if you didn't have any of those that predated on those and you had very highly bacterial soils, well, what would happen then? And this next picture shows us that the trophic levels underground influence plant succession above the ground. If we have very, very highly bacterial soils, we're likely only to get very first succession plants, the pioneer plants, those that come in because they can, that don't need to have such a mature and balanced soil ecosystem. And those are the weeds. So when we think about why we have so many weeds in our soils, and with what we know now, disrupting our soils actually creates the best environment for weeds. We break up the fungal pathways so that the maturity of the soil, the maturity of the soil food web, the trophic levels don't build out as they can because they're always disrupted. And we get those pioneers and those weeds. So we're going to be learning how we can move our soils to growing those trophic levels with as little disruption as possible, yet still able to produce food and crops above the soils. We know that most of our farmers and most of us have seen declining crop yields over time with current agricultural practices, and a lot of that is because we've disturbed the soil food web. So we need to learn to grow with the soil food web. So, for your homework next week, take a walk around your farm and look for fungi or mushrooms. Look in the trees, look under wood on the ground, look in your compost pile, look in your animal manures. Look all over. Wherever you see any decomposing material, you're bound to find fungi at work. Take some pictures and post them in your iCow Soil Biology group, the WhatsApp group. Don't forget to tune in next week for lesson 5, where we'll begin to learn about the third trophic level of microorganisms in healthy soils, the shredders, predators, and grazers. These are the protists, the nematodes, and the microarthropods. We'll start with the protists. In this group we have amoeba, flagellates, and ciliates. So don't forget to join next week to learn about the fabulous amoeba in your soils.
Bye bye for now.